Hello and welcome to my herring dissection video. These are two Pacific herring that I caught here in Sitka Sound a few weeks ago, and they're a little frozen still, so that'll keep them in good shape for dissecting. So we're going to start by looking at some of the external features of the herring. You can see that they've got a really large eye, and they use this eye to feed in low light environments. And here we have the operculum. So that's the flap that covers up their gills. And if we peel that back, we can actually see the gills under there, which is pretty cool. Next, we'll look at their fins. They've got this dorsal fin that comes up right here. No adipose fin. And then this is their tail, or caudal fin. And you can see it's really deeply forked. And then this is the area of the fish called the caudal peduncle, where the tail attaches to the body. And then next, here we have the anal fin, right there. You can see it. Next, there are these two little paired pelvic fins. And then finally, there's the pectoral fin. And you can see these herring have pretty small fins in comparison to their body size. Herring also have scales that really easily flake off. They're very large scales. To look at a herring's coloring, we can see that herring are an example of countershading. So they've got this dark upper body and light lower body and pretty much white belly. This means that if you're a predator looking down, they're going to match the color of the water below them. And if you're a predator looking up, they're going to match the color of the water above them too. They also swim in schools, so that can help protect them through schooling behavior, which makes it harder for predators to capture them. Herring are a really important fish species here in Sitka, as well as around the whole west coast. Their range is from Baja California up through the Bering Sea, and across this range their size varies a little bit, as well as their spawn timing. So herring are considered a keystone species. A lot of the food chain depends on them. Whales eat herring, as well as salmon, and many other commercially important fish consume herring. Now we're going to get into the herring dissection. I have two herring here. I'm going to cut up both of them, and we'll see if they are male or female, or if they've already spawned. Got these fish cut apart, we're going to look at some of their internal features. So I pointed this out earlier, but on this fish we can see the gills really well. I cut away the operculum so you can see we've got the gill arch right along here, and then the gill filaments coming out like this. And then I kind of pulled it out so you can see it a little bit better. We have our heart kind of busted open when I cut it open, so that's why this fish is covered in blood. But yeah, so there's our heart. And so it's located right next to the gills, and it's helping pump blood through the body. Next, you can see we've got our pyloric ceca right here. And this whole section is the digestive system that I'm lifting out right here. And so you can see it has a large amount of surface area, and that will increase the amount of digestion the fish can do. And this fish also had a lot of little parasites, little worms across its digestive system. I put a bunch of them right here, but they're kind of cool, kind of gross, most likely nematodes. And then the digestive system goes all the way down here to the anus right here. So it looks like both these fish are male. You can see these big white sacs of milt. I was kind of hoping we would have a female, but so it goes. So when it comes time to spawn, the fish will all gather in the intertidal zone, or just below the intertidal zone. And the females will release their eggs onto substrates such as kelp and eelgrass. And then the males will also release their sperm from these big sacs. And then the eggs will get fertilized and spend a few weeks on that substrate until the temperature is right and it's time to hatch. Let's look at some other features in here. We kind of have to dig in a little bit. So if we pull all this gunk out, you can see this kind of silvery pouch right here. And I'm pretty sure that is the swim bladder that is currently deflated. So that's pretty cool. Pretty cool to see. Another thing I wanted to mention is that you can see the lateral line on these fish. If we scrape away these scales, you can see right here, you can see it really clearly and all the way along the fish. The lateral line is really important because it helps aid in sensory perception for the fish, so they can kind of feel how the water is moving around them. So that's pretty awesome. So now on this fish, I've cut into the head, and you can see its brain is right here. So if you kind of just cut straight through the head, you can find the brain pretty easily. And the area around it is nice and hard, but the brain itself is quite squishy. Now we're going to see if we can find some otoliths. I've never extracted otoliths from herring before, so we'll see how this goes. So I cut open the head and searched around by the ears and was actually able to find the two otoliths from inside the herring. Here's one of them. Put it right there. Here's another one. I'll also include a picture of those. The otoliths are often used for aging the fish, as with herring, and they're also a unique shape, so you can even use them to ID the fish. Here's my pile of cool stuff that I pulled out, so you can see the otoliths, and then those nematode parasites, and then the eyeball. The otoliths are especially cool. So when you're doing a dissection, it doesn't hurt to just cut open the fish as much as you want. Here I just cut the fish in half so you can see the muscle structure. You can see that these herring are, mostly muscle. They're focusing on just swimming around as much as possible. So here's our muscles, so you can see the lines there, and then the spine is right here where I'm pointing at. So that's pretty cool to see. And then on the inside, you can also see these are their rib bones right along here. So now that I've really cut apart these fish, you can see I've got a bunch of piles of different pieces of the fish. I think we're about done with this dissection. I think it's really awesome once you already have these fish that you're using for science to really get as much out of them as you can. Dissections aren't pretty, but they're pretty fun, and you can learn a lot from them. Thanks so much for watching. Bye.